Typically, we think about life in ideal conditions, where the sun shines and nutrients are rich, but this study is about life that has neither of those. Deep in the ocean, beyond the reach of light, it's so cold that water would be ice if it weren't for the pressure of being thousands of meters below the surface, and hydrothermal vents swing the temperature above the boiling point. This is where we find our sample. Introducing Teskearchaeota, a unique archaea group from hydrothermally impacted sediments of the Guaymas Basin, described here for the very first time by Sophie Hefner, Lacey Levy, and listed co-authors from the Baker Microbial Ecology Lab. Let's start at the beginning. The Guaymas Basin is in the Gulf of California. You see the purple parts? That's 2,000 meters below the surface, and to get there, you need a submarine. This is the Alvin. It's a big deal in ocean science, and Dr. Baker got to take a ride. And here's a picture of the abundance of life they found down there. So on expeditions like this, sediment samples were collected. But these samples are from such an extreme environment, it's nearly impossible to grow them in the lab. Fortunately, we can work with this as long as we can extract the DNA. The DNA from all organisms in the sample is called a metagenome. Through a process called shotgun sequencing, we end up with DNA fragments, which are sequenced, and from there we use a series of computational methods to assemble nearly complete genomes representing the various microbes from the sample. We call these metagenome assembled genomes, or MAGs for short. This means that without culturing the microbe, we can use its MAG for analysis. This is an amino acid identity heat map. It shows us that the Teske group is 99% similar to itself and less than 50% similar to related genomes. To determine the phylogenetic position of our MAGs, we generated this tree using 37 marker genes. Think of key genes like 16S as an example. Notice that the Teske MAGs, highlighted in green, form a distinct clade within the archaeal tax superphylum. Here's where things get really cool. You see, hydrogen as an energy source has been largely overlooked, but according to more recent studies, this abundant resource is ripe for oxidation in the deep sea. Even more interesting, it predates the evolution of photosynthesis, which means we're talking about ancient metabolism. Knowing this, we took suspected hydrogenase genes and generated a tree with more than 2,000 known nickel-iron hydrogenase sequences. This confirmed that our genes fit within these families, and it's likely Teskearchaeota get their energy from oxidizing hydrogen. But this is just the beginning. With evidence that Teskearchaeota are a novel phylum of archaea and may be capable of hydrogen metabolism, our next step is to continue analyzing Teske's metabolism and get a more complete picture of how this organism survives despite its extreme environment.